Okay, good morning. Welcome to Pavitra classes. Today you are studying about the some important enzymes. Some of the enzymes in the biotechnology topic: amylase, catalase, proteases, and penicillinases. These are the some of the enzymes we will study one by one. First one is the amylase. It is an enzyme that breaks starch down into the sugar. So it breaks the starch to the sugar molecules. Amylase was the first enzyme which is discovered by the Anselm Paine in 1833. It is classified as alpha amylase and beta amylase. Two forms are there. One is the alpha amylase and another one is the beta amylase. Amylase is present in the human saliva. In our saliva, in our body, it is present, and it begins with the process of the digestion. So it helps in the food digestion. Usually with the starch, but little sugar contains with the starch, it will break down into the sugar. It helps in the digestion, such as rice, potato. Taste slightly sweet as they are chewed, because amylase turn some of the starch into the sugar in the mouth only. So it's present in the saliva, so it tastes sweet. Because of that enzyme, we feel the sweet taste whenever we eat the things. The pancreas also make the amylase, alpha amylase, to hydrolyze dietary starch into the disaccharides and the trisaccharides. So especially in the carbohydrates classes, amylase enzyme, alpha amylase especially, it hydrolyzes the dietary starch into the disaccharides and the trisaccharides. It is used in the bread making. Usually break down complexes sugar into starch found in flour, so whatever wheat flour or rice flour, into simple sugars. Yeast then feeds on these simple sugars convert into the waste products. So yeasts are usually used for the special microbes as a special microbe production of the amylase enzyme. Waste products specially discovered waste product byproducts are come of alcohols and carbon dioxide. This imparts flavors, causes bread to rise. So after waste products coming into the alcohol and carbon dioxide, this causes the bread to rise. While amylase enzymes are found in the naturally in the yeast cells. So yeast are the microbes used for the amylase synthesis and production. When used as a food additive, this is used as a food additive. It is derived from swine pancreas or mold mushrooms. In that also it will come. Bacillary amylase is also clothing dishwashing detergent to dissolve starches from fabrics and dishes. So bacteria based amylase, bacillary amylase, this gives in the detergent industry, used in the detergent industry, starches from fabrics and dishes also. This is about the amylase enzyme. Second one is the catalase. Catalase is a common enzyme which established in nearly all living organisms. In all organisms it will be present, visible to oxygen, such as plant, animals and bacteria. It catalyzes the decomposition of the hydrogen peroxide. So its main function is it decomposes the hydrogen peroxide to water and the oxygen. It's vital enzyme protecting the cell from the oxidative damages by reactive oxygen species. So it is protecting enzyme. Single catalysis molecule can convert millions of the hydrogen peroxide molecule into the water and the oxygen. So it is present in the living organism. So it converts the hydrogen peroxide to water and the oxygen. Third enzyme is the peroxidase enzyme. So peroxidase HRP is a hemoprotein catalyzing the oxidative by hydrogen peroxide to number of substrates. This also catalyzes, or this also hydrolyzes the hydrogen peroxide to the number of substrates such as ferrocyanide, cytochrome C, ascorbate and leucoform of the several days. So these are the some of the substrates which hydrolyzes the hydrogen peroxide to number of substances like ferrocyanide, cytochrome C, ascorbate, leucoforms in the, and also several dyes. Dye means coloring agent. Peroxide is suitable for the fabrication of enzymes conjugated antibodies, usually in the fabrication enzyme in the antibodies to yield the chromogenic product. Peroxidase characterized immunoglobulins such as immunohistological examination. In the some of the examination processes and medical testing, it is also used to demonstrate the tissue and antigens to quantitative purpose for the soluble and the insoluble enzyme antigens, and in also used in the immunoassays. Next enzyme is the lipase. So lipase is the digestive enzyme. It helps in the digestion of the fatty material. Lipase is used split the fat into fatty acids and the glycerol. So undigested fats, it is splits into fatty acids and the glycerol. Physiological role of the lipases is to hydrolyze the triglyceride to diglyceride, monoglyceride, fatty acids and the glycerol. So the complex fats, it will convert to simple fats. Lipase is produced from the pancreatic seeds, one plant called Euphorbiaceae family, that is Ricinus communis. From this plant also lipase enzymes can be is produced by the in pancreas with the seeds of this plant. And other also, some of the microbes will helps in the production of the enzymes, especially bacteria, fungi and yeast. Because of lower cost and shorter time, optimal growth condition is needed for this. The microbes are used. Lipase production is depend on the number of factors. Usually optimum condition is needed. Carbon, nitrogen sources for the growth as a medium, and pH, temperature, aeration and inoculum size also very important in the lipase production. So what are the strains used in the lipases? Candida lipolytica, Aspergillus niger, Pseudomonas bacillus. These are the some of the fungus and bacteria used in the Lactobacillus sp used in the production of the lipase enzyme. Samples are subjected to serial dilution. So first dilution is takes place. Incubation at 37 degrees Celsius room temperature for around 1 to 2 days, that is 24 to 48 hours. Nutrient agar medium is used. A clear zone around the colonies indicates that production of the lipase. So zone in the colonies, a clear transparent zone indicates that lipase enzyme is produced. So substrates used for lipase enzyme preparation are rice bran, wheat bran, gingerly oil cake, almond meal, mustard oil cake, neem oil cake, groundnut oil cake, olive oil, and as lipid substrate is very suitable for the production of the lipases. Through fermentation, usually with the optimum condition it is prepared. Purification is done later with the centrifugation with treating with the calcium chloride. To stabilize it, calcium chloride is added. After the fermentation, preparation, drying, purification, then centrifugation is done for the filtration, then treating with the calcium chloride. The enzyme is used in yogurt, cheese formation, fermentation, especially in the food industries, conversion of the vegetable oil to fuels, baking industry, dairy industry, chemical industry and pharmaceutical and the cosmetic industry. This enzyme is used. The next enzyme is protease. Protease enzyme is a blend of peptidase. So it has a blend of two enzymes, that is one is the peptidase, one is the proteinase enzyme. To perform the hydrolysis of the peptide bond, it helps in the hydrolyze in the peptide bond. It is a single class of enzyme that has a application in detergent, pharmaceuticals, in the photography and leather and food agriculture industries. Usually Bacillus species, bacteria species is producer of the extracellular protease. This enzyme also used in baking, brewing, meat tenderization, peptide synthesis, medical diagnosis, cheese making and certain of the medical treatment. These are the application of the protease enzyme. So what are the steps involved in the production of the or fermentation of the enzyme? Cultures are deposited in the lyophilized state in the liquid nitrogen. So here liquid nitrogen is used for the production. This is the second enzyme after amylase which has a larger production. Culture are deposited in the liquid nitrogen. Primarily cultures are carried out with the shaken flasks, small with the small fermentation for 40 to 100 m3 or ml at room temperature. Fed batch culture is employed next. So batch culture is employed. Higher oxygen partial pressure is required for the optimum protease. So oxygen is required, aeration required for this fermentation process. It involves around 48 to 72 hours depending on the organism. So which organism we are using, and depending on that 48 to 72 hours it is taking place. Protease is relatively unstable. To stabilize sodium, calcium chloride must be added. So it is unstable. So to stabilize it, the calcium, sodium chloride are added for the stabilizer as a stabilizers to protect that enzyme. Last enzyme is the penicillinase enzyme. So this is a very important enzyme which help in the hydrolysis of beta lactam complexes. So in these 2-3 types are there: beta lactamase 1, beta lactamase 2, cephalosporinase, penicillin amido beta lactam hydrolase. These are the some of the enzymes of the penicillinase subclasses. It is produced from the Bacillus cereus bacteria. periplasmic enzyme with an observed Km of 60 micro μm, that is the benzyl penicillin. So how the production of the penicillinase is take place? In the production of penicillinase, the enzymes are immobilized to the pH sensitive material by cross-linking with the enzyme entrapment method. So many sensors are used for the penicillinase production. So first what they will do, the enzyme is immobilized with the pH sensitive material. So it is a pH sensitive. So cross-linking of the enzyme or entrapment methods for immobilization are used. In this method, penicillinase is immobilized directly to tantalum pentoxide surface. For the immobilization of the enzyme, solution is prepared by dissolving the enzyme in 200 ml of triethylamine buffer at pH 8 of 10 microliter. Enzyme solution per sensor is pipetted. So after this preparation, per sensor pipette is pipetted onto the sample, incubated at the room temperature. So again incubation is done for the 1 to 2 hours. After rinsing and drying with the room temperature, the sensors are stored. So later on it is stored at the multi-component Fulmix buffer to stabilize it, and also at the temperature of the 4 degrees Celsius. This is the one of the method for the penicillinase production of the enzyme. So these are the some important enzymes you have studied. One is the amylase, catalase, peroxidase, lipases, and proteases, and the penicillinase. These are the some of the enzymes which are there in the PCI syllabus 7th semester. So for more classes, subscribe on Pavitra classes. Thank you.